I'm going to speak German. Normally, I would speak English, but I come from Austria originally, and therefore I thought I would speak German. First of all, I'd like to thank the organizers of this meeting. I'm proud of the label, of the name tag that has been given to me. It says artist, and I think that's great. So I'd like to thank Mr. Stocker and his staff. And I'd also like to thank the interpreters. I've never been asked what the title of my presentation is, but I need a title. I call it The Other Memory. We've heard about memory a lot already, but both in humans and in animals, there is an other memory, other than the neuronal memory. I've produced a summary, and I hope in the course of my lecture I will be able to illustrate what I mean. The molecular memory is based in our DNA. The animal neuronal memory is controlled by the expression of the DNA, and it is fixed genetically. The other memory, as I call it, which exists in animals and plants, is controlled epigenetically. I will be talking about epigenetics, and it is subject to environmental influences. Epigenetic changes are much faster than genetic ones. They may be stable over generations, but they are potentially reversible. For the evolution of animals and plants, not just genetics, but also epigenetics is important. All the organisms we know, plants, animals, our snow leopard, or bacteria, have DNA. This is where the genetic information is based. DNA is present in the cell nucleus. What you can see up there in green are the chloroplasts for photosynthesis, and in orange, the mitochondria, which every cell needs to generate energy. DNA is twisted in forms not shown here. Proteins play an important role, and the packaging with the proteins, this is part of epigenetics. DNA, which you already saw when you were invited to this symposium. It's shown here on the cover page of the program. It's a double helix, and I think it's great that artists should have used the double helix as a symbol of science and art. This is DNA as seen under the electron microscope. Incredibly long threads. The length shown in micrometers, millimeters, and meters for different organisms. A virus has a tiny piece of DNA or RNA. A human has DNA which is 1 meter long, 1 millimeter in a tiny cell, constrained in that, packaged into that cell, and it's then wound off. Plants have a broad spectrum and broad range of genetic information. As regards the structure of DNA, it consists of deoxyribonucleic acid, DNA. It consists of the double helix, the sugar phosphate chain and bases shown in 4 different colors, A, T, C, and G. And one base is conditional for the existence of the other colors on the strand, and this is essential for propagation so that the genetic code can be preserved. So complementarity is conditional for that. The violet will get hold of a yellow, and the green of blue. And genetic information is read and propagated that way. So genetics describes the propagation of DNA. This is how the characteristics of humans, plants, animals, and bacteria change over time. That was essential. It was essential for evolution that small errors occurred in the reading of DNA. If that had not been the case, we might still be bacteria today. But as I said, there is also epigenetics. That is a transmission of information that goes beyond genetics. The basis is still genetics, but epigenetics, if you want to explain that in molecular terms, it consists in small changes in the DNA, but it does not change the complementarity of base pairs. It may be a methyl group that plays a role. Small RNA molecules may play a role. The proteins that lead to the twisting of DNA may change in structure and function, and the gene expression is changed that way. I'd like to give you some examples of epigenetics and also talk about how organisms use epigenetics to process environmental influences and to augment these influences. Plants, just like other organisms, are subject to environmental influences. For plants, it is important to propagate correctly, even under such influences, because plants, unlike animals, can't run away if they don't like their location anymore. An interesting method for the transmission of memory consists in the transmission of information between individual parts of a plant. This plant is infested by a pest. That leads to the production of defense mechanisms. The leaf shown in light yellow, in bright yellow. But the leaves that have not been affected and not treated have received the information from the leaf that has been treated that there was an evil around and that they have to defend themselves. This is systemic acquired mechanism. We don't know exactly what kind of information is being transmitted, but the information transmitted is defend yourself. According to some scientists, it even happens between different plants. And it can go on for generations. Something detrimental to the plant is present, be it a virus or UV light or whatever. The plant produces a defense mechanism, but the offspring of that plant that has not been affected still has that defense mechanism. So it's not a genetic change. The DNA is identical, so it must be an epigenetic process. And I'll show you a number of examples in plants and in animals. A classic example was published in 1999 by Agrawal and others. They examined radishes that were infested with pests, a caterpillar. The radish produces certain toxins to defend itself against the caterpillars, and it grows more hair on the leaves and the stem so that the plant can defend itself more effectively against the caterpillar. And the offspring of those radishes will still produce the toxin and produce more and grow more hair. That is an example from literature. Now, an example from my own research. We are doing research on Arabidopsis thaliana. We like that research object very much. It's small, it grows fast, and there are so many mutants of this plant. And we can transform the organism. Genetic transformation is possible. What do? We use a bacterium. We use agrobacteria. That is soil bacteria. A bacterium that is able to modify a plant genetically. In nature, this will produce a tumor because there are genes being transmitted which are important for tumor formation. We can remove these genes and use another gene, a gene for vitamin A, for example. So the new plant, the transformed plant, will then produce vitamin A. Golden rice, for example. You certainly have heard about that. Golden rice that has vitamin A and is more nutritious than normal rice. So the molecular DNA-based memory of these plants has been changed. Another example of an interesting molecular change in plants is the introduction of resistance genes for wheat to make them resistant to certain fungi in varieties that are sensitive. Now, this is being tested in the lab, not yet in the field, but very interesting resistance genes have been found in wild wheat varieties, which are now being transferred to the cultivated varieties. Now, mutations of plants. We've transformed the Arabidopsis plant using a gene which, to detect that gene, we use blue dye. So if the gene is functional, the plant will turn white. If, however, the gene is functional, there will be blue dots, and wherever there is a blue dot, there has been a gene mutation in the gene which we introduced. So this is a test for genetic stability. We use this test, just as an example, to see if Chernobyl and the radiation there is detrimental to plants. And we found that it causes mutation, that it is mutagenic. We were using a living plant to prove the mutagenicity of radioactive irradiation and to show that it's harmful. Not just radiation changes the DNA, also fungi change the DNA. We infected our plant with fungi, and the rate at which mutations occur increases. This is something that was absolutely unknown before. And what's even more interesting, and we only found out by chance years later. Chance, by the way, is important in science. Plants were irradiated and tested in the next generation for spontaneous mutation for the number of blue dots, and the offspring of plants that had been irradiated had more blue dots than plants whose parents had not been irradiated. So plants do have a memory, and the paper we published was called Transgenerational Memory of Stress in Plants. So memory is passed on from one generation to the next, but there is no change in the genetic information in the DNA sequence. And another thing is quite fascinating. A colleague of mine infested tobacco plants with caterpillars, and she tested in the next and the next but one generation to see if the plants had become resistant. And it was not only in the next generation, but in the next but one generation that a certain measure of resistance was found. It ends in the third generation. Wouldn't it be great if we could pre-inoculate a plant one generation before? We wouldn't need any herbicides, no weed killers, if we could do that. Of course, research is not there yet, but it could happen. In my summary, I said that in animals there are mechanisms which we do not always fully understand, but mechanisms by which animals can remember certain influences and pass on to the next generation, not through memory. But through an epigenetic system. In the paper which I mentioned, a paper about the radishes, people were looking at Daphnia. Daphnia are interesting animals living in water, and they have a small helmet. If, however, you release larvae of mosquitoes or small fish attacking the Daphnia, the helm, they grow a bigger helmet so that they can defend themselves, and even their offspring have the enlarged helmets so they can defend themselves, and they become resistant to the larvae of mosquitoes and small fish. And again, it has nothing to do with memory. In a few experiments, it was possible to show that there are epigenetic changes. Here, for example, the research group of Michael Meany, they tested rats, female rats, mothers that show a different behavior with their young. The rats on the left, it's a good mother caring for her children, licking them, stroking them, being nice to them. This can be measured in quantitative terms, but there are other rat mothers who don't care for their offspring, and this is inherited. Now, if the daughter of the bad mother or of the good mother will show the same behavior. Well, that is conceivable in genetic terms, but if you have a rat girl born by a mother who didn't care about their offspring, if you have that rat brought up by a foster mother who is nice and caring, that rat girl, once it has young rats herself, will behave the way the foster mother did. Well, if you look at the data to the left and the right of the rats, you can see how the DNA changes due to a certain receptor protein. The DNA of a certain receptor protein changes. This doesn't mean that this change has its cause in epigenetics. It's hard to establish a causal relationship on this basis. Another example, a fungicide, vinclozolin, has been found to cause fertility defects in the male offspring of rats exposed to the fungicide. The fungicide is used in horticulture, in fruit growing, in vineyards. It's permitted in some countries, not in others, not in Austria, because it's known to cause such changes in rats. These fertility defects have been seen even in the 4th generation of such rats. And it's not quite clear if this kind of research is really reproducible in each and every experiment. Some of the experiments are extremely complicated, and you need to do many, many controls in order to be sure that this is really reliable. Anyway, these effects have been found even in the 4th generation, and even the changed DNA methylation has been found. We don't know if that is a consequence of the changes in behavior. And finally, effects in humans. Of course, you can't experiment with humans. You need to look at data which you find somewhere in the literature. And the research group around Pembrey analyzed the offspring of a small village, Överkalix, in northern Sweden. So they looked at their health conditions, and they found that if the grandparents in the late 19th century, not everybody in the village got the same amount of food, and they found a rather strange correlation. A well-fed grandfather led to an increased mortality in the grandchildren, and a poorly fed grandfather led to a low mortality of the grandchildren. And the controls are absolutely okay. I've seen the data. The figures are not enormous. Of course, there are problems involved because we don't know about the other influences apart from nutrition in that village in northern Sweden. And we don't really know if the grandparents actually ate what there was available. We only know how much food there was available. We don't know if they ate everything they had. So it's a difficult type of research. Because, as I said, you don't experiment with humans, not anymore. Those were the examples I wanted to show you. Back to my summary. As you heard, the molecular memory is based in DNA. The other memory, or the epigenetic memory, I don't know how to call it, exists in animals and plants. It's subject to environmental influences. I gave you the example of the caterpillars, of UV radiation, radioactive radiation, the behavior of female rats. I gave you the example of the fungi and the toxin used against that. So epigenetic changes happen fast. Within a single generation, the defense mechanism is being developed, and it is passed on from generation to generation, from one generation to the next, to the third generation. Even in our own experiment, we've seen 4 generations at most that became resistant to radiation. Epigenetics is a new field of research. There are many unknowns yet to be studied. Epigenetics is everything that's not to do with the primary sequence of the DNA. Small changes in the packaging of the DNA, in the rate at which genetic information can be read in DNA that can be changed by certain proteins or other minute modifications. Well, that's all I wanted to tell you. Questions, I will answer questions later, and I do answer questions by email if you want to send me your questions.